Hello friends, welcome to my course on Microbial Biotechnology. Today we are starting module number 3, and in this first lecture we will be discussing about the basic physiology of microorganisms. This lecture is divided into various sections, 4 sections. So we will start with section number 1, where we are going to give a introduction, then we will move on to metabolism, nutrient acquisition in prokaryotes, then microbial nutrition, particularly the nutritional types, and then the nutrient uptake mechanisms by the passive and active transport modes. So, microorganisms are highly diverse and they can adapt to extreme environments, hot, cold, dry and so on. This is due to their basic physiology that supports their survival, growth and reproduction across varied habitats, and some of these mechanisms by which it is possible are the nutrient acquisition under difficult situations, then the inherent metabolic pathways for energy production and biosynthesis, and then the energy production through respiration and fermentation, and cellular regulation and responses to environmental changes. So, therefore, understanding the microbial physiology is very crucial for advancements in biotechnology, medicine and environmental sciences. Now, briefly, we can describe metabolism as a summation of two processes, the catabolism and anabolism. Both type of pathways are required for maintaining the cell's energy balance, growth and survival. Now, what is catabolism? In catabolism, large molecules are broken down to smaller ones and released energy. While in anabolism, small molecules use the energy to assemble into larger ones. So, they are just the opposite of one another. So, the breakdown of molecules to obtain energy is catabolism. During the catabolic process, complex molecules like carbohydrates, fats and proteins are broken down into simpler ones, releasing energy that is stored in the form of adenosine triphosphate. In anabolism, the synthesis of all compounds are done which are needed by the cell are completed. In anabolic processes, the energy produced from catabolism is used to build up complex molecules like proteins, nucleic acids and polysaccharides from simpler ones. So, catabolism and anabolism are also tightly linked to one another and dependent on one another. So, let us try to understand how the prokaryotes consume food or how they acquire nutrients. Unlike us, they do not have highly developed organs. In fact, they are unicellular. So, microorganisms have evolved the diverse mechanisms of nutrient uptake to efficiently capture and utilize nutrients from various sources. In addition to carbon, hydrogen and oxygen, all organisms require sources of energy and electrons for growth. So, based on these, we can divide microbes into different types, which is basically the nutritional type of the microbes. So, they may be autotrophs and heterotrophs from the point of view of carbon sources. The autotrophs, carbon dioxide is the sole or principal biosynthetic carbon source. For heterotrophs, they use reduced preformed organic molecules from other organisms. So, when you look into the nutritional type from the point of view of energy, we can divide them into phototrophs that use light as their energy source, or they are divided into chemotrophs which obtain energy from the oxidation of chemical compounds, either organic or inorganic. From the electron source point of view, we can classify them as lithotrophs which use reduced inorganic substances as their electron sources, or they may be organotrophs, those extract electrons from organic compounds. So, let us now look into the membrane transport mechanisms. Bacteria and archaea use various methods to obtain nutrients such as passive and facilitated diffusion, active transport and specialized systems like ATP binding cassette transporters. One of the most important functions of the plasma membrane is to control the transport of molecules into and out of the cell. This control is essential to maintain internal conditions within a certain range despite any changes in the external environment, otherwise the organism will perish. Transport of molecules across the plasma membrane occurs in two ways. First way uses energy, and the other is without the use of energy. Let us focus on the pathway which do not use energy, that is known as passive transport, which may be further divided into simple diffusion, where diffusion across the lipid bilayer happens due to a gradient formation of nutrients concentration. And there is facilitated diffusion, where diffusion is facilitated by the presence of certain proteins or certain structures. Then we have the active transport, where we require the hydrolysis of ATP and it uses the proton motive force, and this can be primary active transport or secondary active transport. The primary one, the energy is utilized directly by ATP hydrolysis. In the secondary, energy is utilized indirectly in the form of proton motive forces. So let us look into them one by one. For example, let us start with passive diffusion, which typically refers to the movement of molecules across a membrane from an area of high concentration to an area of low concentration, driven by the concentration gradient. So here we can see the phospholipid bilayer plasma membrane, and then we have this cytoplasm which is inside of the cell, and this is the external area outside the cell. So here in simple diffusion, there is down a concentration gradient directly across the phospholipid bilayer. Small molecules like carbon dioxide may cross the membrane bilayer directly by simple diffusion. In facilitated diffusion, charged molecules like magnesium and large molecules like sugar cannot easily diffuse across the lipid membrane due to their size and charge. Instead, they rely on membrane proteins to help them transport across. We have ion channels and carriers which facilitate the transport of ions like magnesium by allowing them to move through the membrane down their concentration gradients. Then there are carrier proteins or transporters which assist large polar molecules like glucose in moving across the membrane. This process does not require energy and relies on the concentration gradient to accelerate the movement of molecules that cannot diffuse through the membrane directly. So, let us examine what a channel is and how it look like. So, you can see the protein channel over here, and this is the plasma membrane, and here there are certain molecules in the exterior of the cell, and they are moving inside the cell through these into the cytoplasm through this protein cellid. So, this is called as facilitated transport through channels across the membrane. So, from this figure you can see channels are basically membrane proteins with hydrophilic regions that create a hydrated opening, allowing rapid diffusion of specific substances across the membrane. They are having different functions. For example, they enable high speed transport, millions of molecules per second, and they may be gated or open. The open ones allow transport all the time, while the gated ones are regulating the movement of material inside. They may be voltage gated or they may be ligand gated. These channels are having specificity. They are selective, transporting only specific ions or molecules. Some examples are aquaporins that are specialized to allow the fast movement of water, essential for maintaining cellular water balance. Then we have the carriers. They basically bind to substances, triggering a change in shape to facilitate transport across the membrane, specific to a single substance, contributing to membrane selectivity. So, you can see here these carrier protein which is in the plasma membrane, and a particular molecule is binding to it, due to which there is a shape change, and these particular carrier finally delivers the molecule on the into the cytoplasm or inside of the cell. They have limitation in the number of carrier proteins and can affect the transport of necessary material for cell function. Carrier proteins work at a slower rate, 1,000 to million molecules per second. Both eukaryotic and prokaryotic cells use simple diffusion, facilitated diffusion, and active transport for substance movement across the membrane. However, eukaryotic cells have the unique ability to perform endocytosis, where the plasma membrane invaginates to form vesicles or vacuoles, allowing the uptake of large particles or molecules. This process is not found in prokaryotic cells. So, here in phagocytosis, you can see this is the plasma membrane, and these plasma membrane is forming a vacuole here. So, that from the outside of the cell, it can bring in the material by the process of phagocytosis. The cell membrane surrounds the particle and pinches off to form the vacuole, which is internalized along with the material. Then we have the pinocytosis. Here the cell membrane surrounds a small volume and pinches off to form a vesicle. So, they are similar processes, but phagocytosis happens for large particles and pinocytosis happens for small particles. Then we have receptor mediated endocytosis, where uptake is targeted to a specific substance that binds to these receptors on the external cell membrane, and then it again the similar process like phagocytosis and pinocytosis happens, and the material is internalized. But in all these processes we can see that there is a kind of a pinching effect, due to which the vacuole is formed. But in the case of receptor mediated endocytosis, the transport is very, very specific, because if there is no any receptor present for a particular molecule, it will not bind, and this process will, this coated vesicle will be not formed. Now, let us go to the next important mechanism, which is the active transport. Here we have the primary active transport that uses ATP as the energy source to move substances against their concentration gradient from lower to higher concentration. This requires energy because the movement of substances is not passive and goes against the natural tendency of molecules to move from higher to lower concentration. Then we have the ABC or ATP binding cassette transporters. These are a large family of membrane proteins that use the energy from ATP hydrolysis to transport a wide range of substrates across cellular membranes. These transporters are found in all three domains of life: bacteria, archaea, and eukarya, highlighting their evolutionary conservation and essential role in cellular function. So, here we can see a ABC system or ATP binding cassette transporter system. These are protein complexes that use ATP hydrolysis to actively transport a variety of substances across cellular membranes. They are found in prokaryotes, eukaryotes, and archaea, and play a key role in nutrient uptake, toxin export, as well as drug resistance. These transporters are highly versatile, moving a wide range of substrates such as ions, sugars, amino acids, and peptides, and are essential for cell survival in challenging environments. So, you can see here in this ATP binding cassette the ATP binding region. So, here we can see a plasma membrane, and then this is the ATP binding cassette transporter, and this is the ATP binding region, and this is a substrate. This is a substrate binding protein. The substrate binding protein binds to the protein, and then it interacts with the carrier protein, and you can see the substrate is transported inside. And because of the ATP binding over here, there is a conformational change which facilitates the transport of the substrate from the external area of the environment of the cell to the internal cytoplasm. So, basically ATP binding region binds ATP and hydrolyzes it to ADP plus inorganic phosphate. The energy released from ATP hydrolysis is used to drive the transport of the substances. The substrate binding protein binds to the specific substances that is being transported and ferries it to the membrane spanning proteins. Let us now discuss about the secondary active transport. Before that, let us understand some figure as you can see here. Here basically we have a protein, we call it as a uniporter, and these allows the movement of substance or molecule only in one direction, and it allows only the movement of a single species at a particular point of time. Then we have the symporters, where two molecules which are unrelated move together, and their movement may be often coupled. And then we have the antiporter, where the movement of two molecules are opposite to one another. So, when A moves inside, B will be moving outside. So, now the secondary active transport relies on energy from a proton motive force, which is created by electron transport processes. Protons accumulate outside the cell, creating a gradient. Simple transport involves uniport, where single substances are allowed to move in one direction. Then we have the symport, two substances move in the same direction, and antiport, where two substances moves in the opposite direction. This is facilitated by the specific protein porters, which we discussed just a few seconds ago. Then there we have the group translocation, which is a specialized form of active transport that uses energy from an energy rich organic compound instead of ATP. Unlike simple diffusion in ABC transporters, group translocation involves chemically modifying the transported substance during its transport across the membrane. So, here we have these glucose, and you can see here an enzyme, and then the high energy phosphate from PEP you can see over here. So, these diagram basically shows the schematic of group translocation. So, one of the best studied example is the phosphoenol pyruvate sugar phosphotransferase system, which uses energy from PEP. In this system, a phosphate group is transferred from PEP to the incoming sugar molecule. Here you can see this is a high energy phosphate from PEP, and this is the incoming sugar molecule, and here the phosphate group is transferred to the incoming sugar molecule, effectively phosphorylating the sugar during the transport. So, these glucose which was outside, when it is internalized through this mechanism, will be actually phosphorylated. So, this is the glucose 6 phosphate. Let us now move to section 2, where we will be discussing about the strategies for microbial survival and efficiency. Basically, this is done by enhanced transport efficiency or alternative metabolic pathways, then certain special type of relationship we call as symbiotic relationships, or the biofilm formation, endospores and cyst formations, which we have studied earlier. Let us start with the enhanced transport efficiency. Prokaryotes can express high affinity transporters, which are specialized proteins that effectively capture and transport nutrients even when they are present in very low concentrations. This allows them to survive in nutrient poor environments by maximizing the nutrient uptake, and some of the examples are the ABC transporters. Then there are alternative metabolic pathways in response to nutrient scarcity. Prokaryotes can activate alternative metabolic pathways that allow them to utilize different substrates for energy and growth. For instance, when glucose is limited, some bacteria can switch to using other sugars or even lipids and proteins as energy sources. This we will be discussing in detail in later part of this lecture. Some prokaryotes form a symbiotic relationship with other organisms for nutrients, and that may be mutualistic or parasitic, or it may be commensalism. In mutualism, both partners are benefited. In parasitism, one partner is benefited, the other is harmed. in commensalism, one partner benefits, the other is unaffected. So, some of the best example of mutualism is rhizobium bacteria, which fixes nitrogen, atmospheric nitrogen for plants, while plants provide carbohydrates and a nutrient rich environment to the bacteria. Then we have parasites like Plasmodium species, which causes malaria and extract nutrients from red blood cells, depleting host resources and also causing damage or disease to the host. In commensalism, we have the examples of Staphylococcus epidermidis, which is found on human skin. It uses the skin oils and residues as nutrients without significantly impacting the host. Biofilm formation is another survival strategy. Prokaryotes can form biofilms, which are communities of microorganisms embedded in a protective extracellular matrix. So, this is a single bacteria which land on surface, then the bacteria will aggregate and attach, and then there is a microcolony formation of the bacteria, and there is a maturation followed by the dispersal, and then this cycle can go on. So, we call it the biofilm life cycle. These biofilms help prokaryotes survive in nutrient poor environments by concentrating nutrients, protecting against environmental stress, and facilitating cooperation among species within the biofilm. Endospores and cyst formation are also one of the important survival strategies, and here endospores are highly resistant domain structures formed by certain bacteria such as Bacillus and Clostridium. In response to nutrient limitation and other environmental stresses, they are capable of surviving extreme conditions like high heat, radiation, desiccation, and chemicals. So, this is a schematic of an endospore where we can see a core, and then there is a inner membrane and surrounded by a gram cell wall and a cortex, and then outer membrane, and finally a coat. These endospores can remain dormant for extended periods and can reactivate, that is germinate, when favorable conditions such as the availability of nutrients are restored. This process allows the bacterium to survive in harsh environments and resume growth when conditions improve. We have already discussed about these in details in module number 2. Then there are the cysts, which are also dormant protective structures formed by certain microorganisms to endure nutrient scarcity and environmental stresses. Cysts typically have thickened walls that shield the organism from harmful conditions such as desiccation or chemical exposure. Like endospore, cysts remain dormant until environmental conditions turn favorable or improve, at which point they can reactivate and resume normal activity. So, you can see here the cyst formation in Rhodospirillum centrum. Let us now move on to section 3, where we will be discussing the metabolic processes, mainly the various aerobic respiration processes and the anaerobic respiration processes. So, what is aerobic respiration? Aerobic respiration is a process that extracts energy stored in glucose or other organic molecules in the presence of oxygen, producing ATP, carbon dioxide and water as byproducts. This process can be represented by the following chemical equation, where glucose utilizing oxygen to form carbon dioxide, water and energy in the form of ATP. The breakdown of glucose through aerobic respiration yields approximately 2900 kilojoules per mole of energy, which is used to produce ATP molecules that power various cellular functions. The actual energy yield per glucose molecule is around 30 to 32 ATP, and this is one of the most important source of energy for the cell. So, let us look into the various steps involved in the aerobic respiration. The first and foremost is the glycolysis, which is the initial step of aerobic respiration. Glycolysis occurs in the cytosol of the cell. During this process, glucose molecules are converted to pyruvate, resulting in the formation of 2 ATP and 2 NADH molecules, which are later utilized in aerobic respiration. Then we have the Krebs cycle. During this stage, oxaloacetate combines with acetyl coenzyme A to generate citric acid, a series of reactions resulting in the production of 2 molecules of carbon dioxide, 1 molecule of ATP and a reduced form of NADH and FADH happens. Then in the final step of aerobic respiration is the electron transport chain. In this phase, a significant amount of ATP is generated through the transfer of electrons from NADH and FADH. A molecule of glucose yields a total of 34 ATP molecules. So, if we look into aerobic respiration in a nutshell, so this is the cell membrane which delineates the external and internal environment of a cell, and then glucose, which is the life's basic nutrient, enters the cell by various transport mechanisms, and it undergoes glycolysis. The energy handling process for all of life forms have many common elements. So, these glucose undergoes glycolysis, and it produces some ATP molecule, 2 ATP molecules, and also it is converted to pyruvate, which in the absence of oxygen will undergo fermentation and produce lactate. And in the presence of oxygen, it will be converted to acetyl coenzyme A, transported into the mitochondria, where it enters the TCA cycle, and from here these NADH and FADH2 enters the electron transport, which produces many ATP molecules down the line. So, this is life's basic nutrient, and this is the life's energy factory, which is the mitochondria, which produces the ATP, which is the life's fuel. So, we will be discussing many of these processes as well as the fermentation process towards the end of this lecture. So, let us start with glycolysis, which is one of the most important metabolic pathway. This is also called the Embden-Meyerhof-Parnas pathway, or commonly known as the glycolysis, a primary pathway for glucose catabolism in both prokaryotic and eukaryotic cells. It occurs in the cytoplasm and can function under both aerobic and anaerobic conditions, though it does not require oxygen to proceed. So, basically glucose is converted to pyruvate, where it may undergo oxidative phosphorylation, or it may undergo anaerobic pathway, which is the fermentation pathways, as we have shown in the earlier picture. Then there are alternate pathways like the pentose phosphate pathway and Entner Duodorov pathway, which are alternative pathways to glycolysis. They play crucial roles in cellular metabolism, especially in the prokaryotes. The pentose phosphate pathway is involved in generating NADPH and ribose sugars for biosynthesis, while the Entner Duodorov pathway is important for energy production in some bacteria. Both pathways intersect with glycolysis at key intermediates. Here we can see a comparison between the EMP pathway, the PPP pathway, and the ED pathway from the point of view of primary function. The EMP pathway, the breakdown of glucose to produce ATP, NADH and pyruvate. In PPP, it generates NADPH and ribose 5-phosphate for biosynthesis. In ED, this is an alternative glucose breakdown pathway which produces ATP, NADPH and pyruvate. The EMP pathway is common in both prokaryotes and eukaryotes. PPP pathway also found in both prokaryotes and eukaryotes. ED pathway found mostly in prokaryotes like Pseudomonas and Enterococcus. Then the key products of EMP pathway, we have 2 ATP, 2 NADH, 2 pyruvate per glucose. The key product of PPP is 2 NADPH, ribose 5-phosphate and intermediates for biosynthesis. Then we have the products of ED pathway, 1 ATP, 1 NADPH, 1 NADH, 2 pyruvate per glucose. The metabolic role played by the EMP pathway, energy production and precursor for biosynthesis. In PPP, it is biosynthesis of nucleotides and amino acids. In ED pathway, it is energy production and precursor for biosynthesis. The significance of EMP pathway is it is a primary glycolytic pathway in most organisms. PPP provides precursors for nucleotides in biosynthesis and reducing power, and ED here it is used by bacteria that lack certain enzymes for the EMP pathway. Why the alternative pathways are important? It is important from the point of view of adaptability. Both the PPP and ED pathways can intersect with glycolysis, feeding intermediates into or out of the glycolysis depending on the cell's metabolic needs. Then it offers flexibility in metabolism. The existence of these pathways allows cells to adapt to varying environmental conditions, optimize energy production, and generate essential biosynthetic precursors. They also help in regulation of carbon flow. Cells can regulate the flux through glycolysis, PPP, and ED pathways depending on the availability of glucose, the need for NADPH, and the demand for anabolic precursors. Let us now move on to the next important cycle, which is the TCA cycle or tricarboxylic acid cycle. This is also known as the Krebs cycle. Here it oxidizes acetyl coenzyme A from amino acids, fatty acids, and carbohydrates producing NADH, FADH2, and ATP. In prokaryotes, it occurs in the cytoplasm, while in eukaryotes, it takes place in the mitochondria. This is the schematic of the TCA cycle. So, these pyruvate enters, and then it is converted into acetyl coenzyme A, which enters the TCA cycle and produces NADH, then FADH2 and ATP. Then comes the electron transport and energy generation. The ETC is responsible for the majority of ATP production in the cell. The electron flow through the chain transfers electrons to oxygen and pumps protons across the membrane. This creates an electrochemical gradient. The return of protons through ATP synthesis drives ATP synthesis generating cellular energy. So, these ETC has various protein complexes. It is located in the plasma membrane of prokaryotes and the mitochondrial inner membrane of eukaryotes. It consists of 4 proteins, 1, 2, 3, 4, and those are named as complex 1, 2, 3 and 4. Complex 1 is NADH ubiquinone oxidoreductase, an alternate name is dehydrogenase. Complex 2 is succinate dehydrogenase. Complex 3 is cytochrome BC1 complex, and complex 4 comprises of cytochrome C oxidase. Both the prokaryotes and eukaryotes use mobile electron carriers like ubiquinone and cytochrome C to transfer electrons through the chain. This process creates a protein gradient across the membrane which drives ATP synthesis through ATP synthase. We will try to look into the structure and function of ATP synthesis at a later point of time. The electrons flow through the complexes and helps pump protons across the membrane, which is essential for generating the energy needed for ATP production. So, let us see the role of complex 1 or NADH oxidoreductase. NADH donates 2 electrons to complex 1, leading to pumping of 4 hydrogen ions across the inner membrane. NADH is oxidized to NAD plus and recycled back into the Krebs cycle. Complex 1 transfers the electrons to ubiquinone Q, which is reduced to ubiquinol or UH2. Ubiquinol carries the electrons to complex 3. In complex 2, we have succinate dehydrogenase. FADH transfers electrons to complex 2, which are then passed to ubiquinone. Ubiquinone coenzyme shuttles electrons from complex 1 and 2 to complex 3. It is a lipid soluble carrier that diffuses within the plasma membrane. Ubiquinone Q is reduced to ubiquinol QH2, which carries the electrons to complex 3. So, complex 3 is basically cytochrome BC1 complex. The electron transfer to complex 3 facilitates the transport of 4 more hydrogen ions across the inner membrane. Ubiquinol is oxidized and the electrons are transferred to another electron carrier protein called cytochrome C. So, this is basically the part of the complex 4, where cytochrome C oxidase is the main player. Cytochrome C transfers electrons to the complex 4, which pumps protons across the inner mitochondrial membrane. The electrons are then passed to oxygen, which splits and combines with hydrogen to form water, as you can see over here. This process is crucial for continuation of the electron transport chain and for cellular energy production. Cytochrome C is a mobile electron carrier transfer. Electrons from complex 3 to complex 4. In prokaryotes, it is often membrane bound or loosely associated with the membrane. Let us now study a little bit about the ATP synthase. This is the structure of ATP synthase, which is basically a membrane bound protein. So, this is the membrane and this is the cytoplasmic side and this is the external side. Okay. Now, ATP synthase is a membrane bound enzyme complex that synthesizes ATP from ADP and inorganic phosphate using energy from a proton gradient. In eukaryotes, it is located in the inner mitochondrial membrane, while in prokaryotes, it is found in the plasma membrane. ATP synthase functions as a molecular motor, playing a crucial role in cellular energy production. Let us now discuss briefly about anaerobic respiration. Anaerobic respiration occurs in the absence of oxygen, where microorganisms are forced to use alternative terminal electron acceptors, and those acceptors may be nitrate, sulfur, sulfate or carbonate, and through these they generate energy. While anaerobic respiration is less efficient than aerobic respiration, it allows organisms to survive and produce energy in oxygen depleted environments. So, based on the terminal electron acceptors, these anaerobic respiration can be named as nitrate respiration, where nitrate is used as the terminal electron acceptor, or it may be sulfate respiration, where sulfate serves as the electron acceptor, or sulfur respiration, where sulfur is reduced to hydrogen sulfide. Then it may be carbonate respiration, where carbonate is used as the electron acceptor. Then we have other terminal electron acceptors. In case of certain bacteria, they can also use ferric iron or manganese and various other organic compounds, example humic substances, as terminal electron acceptors in anaerobic respiration. Now, let us move to the last section of this lecture, which is the fermentation. Briefly, we have various kinds of fermentation, which is alcoholic, lactic acid, mixed acid, 2,3-butanediol, propionic acid or butyric acid fermentation. Let us try to understand what fermentation is. Basically, when respiration is not possible, organisms must use fermentation to regenerate NAD+, which is required for further catabolism. Fermentation occurs entirely in the cytoplasm, involves no ETC, and uses an organic molecule such as pyruvate as the final electron acceptor, producing reduced waste products like alcohols and acids, and these are actually valuable commercial products. The process is inefficient in terms of energy recovery from glucose, as little or no ATP is formed, but from the product point of view, products like alcohol and acids, it's actually very commercially important. So, in alcoholic fermentation, we have organisms like yeast, filamentous fungi, and certain bacteria which carries out the alcoholic fermentation. So, here you can see glucose is getting converted to ethanol in the presence of 2 ADP and 2 inorganic phosphate, and also producing carbon dioxide in the process. ATP is also produced here overall, but this ATP is not produced during the fermentative step. It was actually produced early in the glycolysis phase, where glucose was broken down. This is basically a 2-step process. Pyruvate from the EMP pathway or ED pathway in gymnomonas is decarboxylated to acetaldehyde. NAD+ is regenerated during the reduction of acetaldehyde to ethanol. Then we have the lactic acid fermentations, where we have various bacteria like Streptococcus, Lactobacillus, Lactococcus, leuconostoc, fungi, algae, and protozoa, which converts glucose in the presence of ADP and inorganic phosphate to lactic acid. And here also ATP is produced, but this ATP is not produced in the fermentative step. It was produced early in the glycolysis phase. Here again pyruvate is the electron acceptor forming lactate. There are two forms. Homolactic fermentation, example Lactobacillus acidophilus, reduces most pyruvate to lactic acid. Then there is a heterolactic fermentation, as that carried out by Leuconostoc mesenteroides, which generates various products including lactate, ethanol, and acetate. Now we have mixed acid fermentation. These are carried out by E. coli and related facultative anaerobes. Products include lactate, acetate, small ethanol quantities, and formate. Some organisms can further reduce formate to hydrogen and carbon dioxide. Then we have the 2,3-butanediol fermentation performed by Enterobacter, Erwinia, Klebsiella, and Serratia. This is similar to mixed acid fermentation, but generates butanediol along with the ethanol and the acids. Then we have the propionic acid fermentation. This is mostly carried out by gut bacteria like Propionibacterium. Some are involved in Swiss type Cs and vitamin B12 production. Propionate is formed from pyruvate via the methyl malonyl coenzyme A pathway involving carboxylation and reduction. Then we have the butyric acid fermentation, which is carried out by Clostridium species, which are anaerobic spore formers. Products include butyric acid, acetone, butanol, propanol, other alcohols and acids. These bacteria also ferment amino acids, nitrogenous compounds, and carbohydrates. So with this we come to end of this lecture number 1 of module 3. Thank you for your patient hearing.